Now we are going to study about the immunoblotting technique. What is immunoblotting technique? Immunoblotting is a specific tool or specific analytical tool which is used in order to identify desired DNA or RNA. So if we want to identify our desired DNA or RNA, we use this immunoblotting technique. Identification of desired DNA or RNA fragment is nothing but we can do this by immunoblotting technique. So what is blotting? Blotting is a method used for immobilization of nucleic acid or protein with the help of solid matrix. Some examples of solid matrix are nylon, polystyrene, nitrocellulose. If we immobilize nucleic acid or protein with the help of a solid matrix, then that method is known as blotting. There are three different types. Actually, there are different types of blotting. Three different types of blotting: Western blotting, Southern blotting, Northern blotting. Western blotting is used for the detection of proteins, while Northern blotting is used for the detection of RNA. Southern blotting is used for the detection of DNA. We can also detect protein with the help of ELISA. Now, what is ELISA? For the detection of DNA, ELISA is mostly used because it is mostly preferred for its sensitivity. Now we are going to see the principle involved in the blotting. Actually, there are 4 steps involved in the blotting. Firstly, we need to extract the proteins or nucleic acids from the electrophoretic method, and we should transfer the proteins or nucleic acids and immobilize them on the paper. Now, to the immobilized proteins or nucleic acid, analytical probe is bound to the probe, and the brown probe undergoes visualization. So these are the steps. First is electrophoretic extraction of protein or nucleic acid, transfer and immobilization on the paper, binding of analytical probe to the target, bound probe visualization. Proteins or nucleic acids are separated from the cell by using electrophoretic method in the electrophoretic extraction of protein or nucleic acid. Then these proteins or nucleic acids are fixed to a solid matrix. So this process is immobilization, and then to these immobilized proteins or nucleic acids, analytical probe is bound to the target. Now, analytical probe is a small fragment of DNA or RNA. This bound probe is visualized under using spectrophotometric method. The bound probe is nothing but colored product, so when we observe in a spectrophotometric method, we can see it. Now, ELISA technique. ELISA or EIA. What is ELISA? ELISA is enzyme-linked immunosorbate assay. It is also called as enzyme immunosorbent assay. EIA, enzyme immunosorbate assay. Now, ELISA is also called qualitative or quantitative assay. ELISA is a solid phase EIA. So, solid phase EIA is known as ELISA. Mostly we use ELISA for diagnostic purpose. There are 3 different types of ELISA: indirect ELISA, direct ELISA, competitive ELISA. In indirect ELISA, it is used for the detection of presence of antibodies. Direct ELISA is used for the detection of presence of antigens, while competitive ELISA is used for the detection of the concentration of antigen. Indirect ELISA is used for the presence of antibodies. It is used for the presence of antigens. It is used for the detection of concentration of antigens. Now, the principle involved in ELISA is, in a well, a probe is taken and in them primary antibodies are added first, and then antigens are added. Antibodies and antigens are reacted, and then secondary antibodies are added. This secondary antibody is enzyme labeled antibody. This reacts with the antigen, and then a substrate is added. So this is the principle. We should take a well with probes. Probes is nothing but small attaching material, and primary antibodies are added. Then a sample containing a concentration of antigen is added. This is the procedure. Sample containing a concentration of antigen is added. The antibodies react with the antigen. After this, washing is done to remove the excess antigen. Then one more secondary antibody is added. This is an enzyme labeled antibody. This enzyme labeled antibody reacts with the antigen and reacts with the antigen. Then a substrate is added. This added substrate is chromatographic in nature. Chromatographic is nothing but it contains color. So enzyme labeled antibodies react with this substrate to form a product. And the formed product is in the colored form. Since substrate is also chromatographic and colored form, formed product is in colored form. So, and then it is visualized using spectrophotometry. Visualize means it is seen using spectrophotometry. The advantages of ELISA are: the diagnosis is accurate. It is helpful for the carrying of complex sample. We can perform ELISA to a complex sample. Accurate diagnosis can be done. It is highly responsive, rapid and quick results are obtained. The process is easy and uncomplicated, highly sensitive, and there is no exposure to the radiation. Quantitative, semi-quantitative, qualitative. Calibration, standard curve methods can all be used for the detection. So for the detection of proteins, quantitative, semi-quantitative, qualitative, calibration, standard curve methods can be used for the detection. So these are the advantages. I will revise it again. Acute diagnosis can be done. It is highly responsible. Rapid and quick results are obtained. It is the process is easy and uncomplicated. It is highly responsible. No exposure to the radiation. Complex sample can also be performed. We can also perform ELISA to complex sample. And for the detection, quantitative, semi-quantitative, qualitative, calibration, standard curve methods can be used. Now, after the advantages, we will study about the applications. For the applications of ELISA are, as we saw, we can detect the presence of antigen and the concentration of antigens, and it is used for determination of serum antibodies. It is used in the food industry in order to detect allergens. We can track detection. We can also detect spreading organisms like viruses like HIV virus, Zika virus, rotavirus, organism causing bird flu, cholera, STDs, flu. These all organisms can be detected using ELISA. Applications are for the detection of antigen antibody, determination of serum antibodies, and then used in the food industry for detection of food allergens. We can also detect the number of spreading organisms like HIV viruses, Zika virus, rotavirus, organism causing bird flu, and the cholera, STD, flu. All these can be detected. Now we will revise again from the starting. What is immunoblotting technique? Immunoblotting technique is a specific tool or an analytical tool which we use in order to identify desired DNA or RNA. To identify desired DNA or RNA, we use this technique, it is a specific tool or an analytical tool. Now, this is the definition of immunoblotting technique. Now, what is blotting? Blotting is nothing but we immobilize for the immobilization of nucleic acid or protein with the help of solid matrix is nothing but blotting. Immobilization of nucleic acid or protein with the help of solid matrix by using a solid matrix is known as a blotting method. Example of solid matrix are nylon, polystyrene, nitrocellulose. These are the examples of solid matrix: nylon, polystyrene and nitrocellulose. There are three different types of blotting: western blotting, northern blotting, southern blotting. For the detection of protein, we use western blotting. For the detection of RNA, we use northern blotting. For the detection of DNA, we use southern blotting. For the protein detection, we also use ELISA. Actually, ELISA is mostly preferred because of its sensitivity. Now, principles involved in the blotting: electrophoretic extraction of proteins or nucleic acids, transfer and immobilization on the paper, binding of analytical probe to target, bound probe with visualization. Now, proteins or nucleic acids are first extracted using electrophoretic method. Now, the extracted proteins or nucleic acid are fixed to the solid matrix. This is immobilization. And then analytical probe is bound to the target. Analytical probe is nothing but a small fragment of DNA or RNA. We should visualize the formed bound probe using spectrophotometric method. The bound probe is colored product, so we can identify or detect. Again, ELISA. What is ELISA? Enzyme linked immunosorbent assay or enzyme immunosorbent assay. Solid phase EIA. Solid phase EIA is ELISA. ELISA is also known as qualitative or quantitative assay. We use ELISA for diagnostic purposes. There are three types of ELISA: indirect, direct and competitive. Direct ELISA is also called as sandwich ELISA. Indirect ELISA is helpful for the detection of presence of antibodies. Firstly, it detects presence of antibodies. Direct ELISA, also known as sandwich ELISA, it is used further to know the presence of antigens, while competitive ELISA is used for the detection of concentration of antigens. The principle involved in ELISA is nothing but we should take a well with probes and then primary antibodies are added. Then a sample containing a concentration of antigen is added. Antibody react with the antigen. Antibody react with the antigen, forms a complex, and then it is washed. Washing is done to remove the excess antigen and secondary antibodies are added. These secondary antibodies are enzyme labeled antibodies. Reaction between antigen and enzyme linked antibodies is done, and then a substrate is added which is chromatographic in nature. Chromatographic is nothing but it contains color. Substrate reacts with the enzyme labeled antibody, which forms a product. This product is in colored form and then it is visualized using spectrophotometer. The advantages of ELISA are accurate diagnosis can be done. We can perform ELISA using complex sample. Accurate diagnosis can be done. It is a simple, easy, uncomplicated process. It's highly responsive. It is highly sensitive, highly responsive, and no exposure to the radiation. It gives rapid and quick results. For the detection, quantitative, semi-quantitative, qualitative calibration, standard curve methods are all can be used. Now, applications. The applications are detection of antigen and antibody, determination of serum antibodies, used in food industries for detection of food allergens. We can track or detect number of spreading organisms like HIV virus, STDs, flu, bird flu, organism causing bird flu, cholera, Zika virus, rotavirus, etc. Thank you.